Hello, everyone. Welcome to our webinar, New Developments in Genetic Knowledge for Defendants in Asbestos Cancer Cases. For those of you who signed in 2 weeks ago, we apologize for any inconvenience we caused due to the technology not working properly. So our goal today is to show you the power of genetics in toxic tort cases. We'll provide you with new information to make the case that mesothelioma or ovarian cancer in some individual plaintiffs was caused by that person's genetic predisposition, not by asbestos or talc. Our organization is called ToxicoGenomica. We are a strategic alliance of scientific consultants, geneticists, and legal professionals. Our goal is to facilitate the use of genetics in toxic tort cases and to make it understandable and accessible to the legal profession, from the lab bench to the courtroom. I am David Schwartz, a founding partner at Innovative Science Solutions. Kirk Hartley, founder of the LSP Group, is a litigator with over 30 years of experience defending mass tort litigation, including many asbestos cases. Len Van Zyl is the founding CEO of Array Express, a laboratory that performs genetic sequencing. Dr. Van Zyl is a PhD geneticist who has been offered as an expert witness in dozens of toxic tort cases and has even testified at trial in asbestos cases. Len has played a major role in demonstrating that cancer in individual plaintiffs was caused by their genetics, not by exposure to an alleged toxicant. Here's an overview of what you'll be hearing over the course of our webinar this afternoon. Kirk is going to start by giving you an update on the use of genetics in legal cases around the country. Len will then provide you with a very short overview of genomic science basics. He will show you how this information can be applied to demonstrate that genetic mutations are the cause of cancers relevant to asbestos and to talc litigation, specifically mesothelioma and ovarian cancer. Finally, we'll describe our custom gene panel tests designed specifically for busy litigation teams, including how to pick individual cases most likely to yield positive outcomes in the courtroom. Please note that the slide presentation, as well as our informative white paper, is available to download from your GoToWebinar side panel. After the presentation, you'll receive a short survey that we hope you will complete to give us feedback on the webinar. So with that, let me turn it over to Kirk Hartley.
Thank you, David. Good afternoon, everyone. I'm going to start with an overview, big picture points. The general rule is that courts are allowing genetic testing when it's requested by defendants, and in some cases, plaintiffs are even acknowledging the reality and going along with it. There was one contrary ruling in the Marshall case in California. That was a situation in which the defendants did not use an actual genetic expert, and I think that can be overcome. Note that Mark Lanier and other plaintiffs' lawyers are expanding their use of the concept of susceptibility. We've seen that in both asbestos cases and in the Ingham trial involving ovarian cancer. There they very much focused on susceptibility. Note also that the Pinal paper, which came out in early August of this year, it makes frequent use of the term susceptibility. Perhaps not so coincidentally, one of the authors is Dr. Hetty Kindler, who most of you will know has tight ties to Cooney and Conway. And I think you need to be aware of that paper and its frequent use of the term susceptibility. We've done a blog post about that gets into more detail. The big picture is that between the Pinal paper and others, more and more genes are being associated with the development of mesothelioma. And so just thinking about BAP1 is not enough. You need to be thinking about multiple genes. Next, please. Sorry, go back one. Another development we're seeing is that Dr. Testa, who's testified for plaintiffs in a couple of the cases, has ongoing mouse studies involving knockout mice that are being exposed to asbestos, including chrysotile. You need to be aware of that. There's interim data out as of May that's not published. More data should be coming out later this year or early next year. There's also some instances in which the smoking is becoming relevant to cases. We'll talk a little more about that. Next, please. David mentioned that Len has testified in asbestos cases. That's correct. David misspoke in saying he testified at trial. The key mesothelioma case in which Len testified was the Blackford-Cleaton case, which is highlighted in the middle of this chart. That was a case brought by the Simmons firm. Ultimately, it turned out that that woman had a mutation in a gene known as CDKN2A, which is tied to efforts to repair double strand breaks, which can arise from smoking and access to sun tanning, both of which she did. In our view, that provided a powerful defense, and our client reported a good but confidential outcome. Genetics also have been used by Len and us in other cases, so you realize that this is not just about asbestos. In the benzene cases, Len's been involved in multiple situations where genetic testing was able to be used to show that a person was destined to develop cancer. And then in the Guzman case, he was able to use gene expression testing to show that a papillary thyroid cancer had the fingerprints relevant to a genetically caused tumor as opposed to a tumor arising from exposure to radiation. Next, please. With respect to the embrace of susceptibility, some of you may have seen this drawing from the Ingham trial in articles that are circulating. This is, in fact, exactly what Lanier used at trial. Ultimately, they took the viewpoint that susceptibility could be found in genetic variations and other risk factors, and they embraced it, and they used an expert to try to simply embrace the whole idea. And so our point is we believe defense lawyers need to be prepared for this and think about how to deal with experts both in deposition and at trial. And in that case, they actually used a very well-credentialed Stanford oncologist who's an MD, PhD. Next, please. We already covered this. Let's go by it, please. Next, please, David. So a specific example of the outcomes, the Nolan Lam case, is one in which BAP1 was nominally at issue. From what I understand from different people who were involved in the trial, the verdict really was based on other factors. The Marshall case I mentioned and is the one where Kazin prevailed in opposing genetic testing. He's gone on to make a YouTube video about it. The link is at the bottom of the page. If you're up against Kazin in a California case, you should be aware of that. Next, please. Other examples of where it's been done, the Lonzo case in New Jersey. There's a CMO pending out there seeking genetic testing. Genetic testing also was approved and then used in the Bailey case in Lawrence County, Tennessee. Next. We would urge you to be aware that there's a broad look at the science as related to low dose and mesos in women in the Bestwall Chapter 11 bankruptcy. We expect that genetics are going to be argued in that case. There, the plaintiffs' firms have 4 different plaintiffs' firms have been added to the science team to take on the issues. Right now, it's in motion practice, but assuming the case is not knocked out on motion practice, I think you can expect to see some of these low dose and genetic issues litigated there. We're following it and we'll be continuing to write about it. Next. For those of you who have not seen it, I wanted to point out that Suresh Mulgivkar, who many people have seen used as a defense expert in asbestos cases, has acknowledged that individual variability arising from gene defects is in fact a significant factor in the cancers in some people. He's acknowledged that the existing cancer models don't acknowledge individual variability because the techniques just weren't there. And he's also pointed out in the Testa treatise that smoking as a cause was missed because epidemiology, is too blunt a tool. And that correlates to some of the things that Len had to say in the Blackford-Clayton case. Next, please. So at the end of the day, the big picture is that, in fact, each person is unique. You can see that when genetic testing is applied. We are trying to make that easier. Some key facts to look for are early onset cancer, multiple primary cancers in the plaintiff, and or a case where you just can't find a dose. Now, genetic investigation is like forensic investigation. Sometimes, you just can't guarantee exactly how things are going to be turning out. However, using good scrutiny early on and looking at the family history and the facts of the particular plaintiff through medical records, it's generally possible to make a good assessment of what's reasonably likely to occur if genetic testing is pursued. But it's not for use in every case, and we wouldn't suggest it's for use in every case. And then last, we believe that a value of this is that it offers juries something objective to hang their hat on and reach a not liable verdict. And that occurred in the Guzman case, which went to verdict in Louisiana and resulted in a not liable finding when Len used the objective molecular data to indicate that it was a genetically caused tumor and not caused by exposure to radiation. So with that, I'll turn it over to Len.
All right. Good afternoon, everybody. Everybody probably heard this term, cancer is a disease of the genome. At the molecular level, that's exactly what it is. All cancers, and I emphasize all cancers, are caused by accumulation of genetic lesions. Now, the lesions can be point mutations, it can be insertions, it can be deletions in the DNA, it can be gene fusion products or chromosomal translocations. Nonetheless, whatever it is, it causes the cells to disrupt their normal cellular processes. Every cell in our body has a predestined stem cell division. It matures, it differentiates into the final tissue, and then it's programmed to naturally die. All those phases of stem cell division and maturation and differentiation is governed and controlled by genes. We refer to those as checkpoints. So it's an extremely robust system, and we have checkpoints to make sure that these cellular processes proceed as programmed in the genetic blueprint. And we also have guardians of genes that guards the genomic integrity. Now, when a mutation happens within those key checkpoint regulators, that's when we start seeing cells to behave differently and turn on to malignant cells that controls uncontrollably. Next slide. Now, how do we get these mutations? And today we're going to focus particularly on mutations as to genetic predisposition. Now, we can either inherit these mutations from mom and dad. Now, when you inherit a mutation from mom and dad, we refer to those as germline mutations. The key point here is that mutation is present in every single cell of your body, and every day, trillions of stem cell divisions happen to maintain us as human beings. Every time a cell divides, the entire DNA copy is copied, a copy is made and transferred to the daughter cells that then differentiate into the final cell. Every mutation that is present at birth is then copied, and typically what's associated with people that inherited a pathogenic mutation, they, whenever the DNA is copied during stem cell division, introduce more and more mutations. And that's why you typically see people that have a genetic predisposition to a disease or to cancer typically develop it more earlier in their lifetime. Somatic mutations, on the other hand, are mutations that we acquire. Cancer is a disease of the aged. Cancer normally occurs late in life and usually is acquired due to either our checkpoint mechanisms that becomes less fit as we age, and errors does slip through during when the cells are divided and the DNA is copied. And that's why typically in somatic mutations that drives the onset of many tumors later on in life. The point here is these mutations are tissue specific. That's why you have breast cancer, lung cancer, kidney cancer, and they cannot be passed on to the next generation. Next slide. So what's really important for us in toxic tort litigation is the issue, and we constantly are confronted with this, whether it's genetic predisposition or susceptibility. And I will try to kind of parse out why these things are actually way different from one another. Many diseases and conditions arise from inherited mutations that we inherit from mom and dad. We are well known with sickle cell anemia, cystic fibrosis, Huntington disease as single gene mutations that cause these diseases. Now, cancers are not different. There are currently more than 200 known hereditary, or sometimes referred to as familial, cancer predisposition syndromes. This implies that for each of these cancer syndromes, there are a series of gene mutations that causes this cancer. They are not risk factors, they are causing the cancer, and they are inherited in Mendel's segregated manner, either autosomal dominant or autosomal recessive. But the point I want to make here that these mutations are known to initiate tumorigenesis, and that can be done absolutely in the absence of a toxicant. Every single cell of a person's body has that mutation if they are genetically predisposed. And when they will develop the disease depends on the penetrance of that mutation. Not all mutations are created equally. Some mutations have a fatal flaw to the protein for which the gene is coding, and others might change just slightly the function of the protein. But nonetheless, they will cause the disease sometime in a person's life. And that is a very important aspect that defense counsel should consider, that many cancers, in fact, all cancers, are now known to have a pure familial or hereditary predisposition to develop that disease. Next, please. The majority of us on this field call will never develop cancer. In fact, estimates are that less than 1% of people develop cancer. Cancer is a rare disease. Most of us are actually, due to evolutionary development and conservation over millions of years, have developed incredibly robust systems. These checkpoint mechanisms are incredibly robust to ensure that we maintain a solid genetic integrity, that our genome is stable under normal circumstances. That's why most of us are actually develop mechanisms that maintain genomic integrity, that maintain us to be healthy to the most part. And that's a very important aspect that defense strategy need to focus on, is that the majority of people have genetic blueprint that actually protects them, that guards them to develop these mutations that's associated with the onset of disease. And with most cases, we understand which those mutations are, and we can determine and look for them in subsequent studies. Now, genetic resistance, next slide, or susceptibility is what we are constantly confronted with. Susceptibility is actually a very complex concept. It is what's coded in our genome, and how that interacts with what we are doing in our lifestyle choices, whether we are active, whether we smoke, whether we drink, whether we have diabetes, whether we have an autoimmune disease, and then, of course, also the environment we live in. It's not a simple, straightforward strategy. However, the point what I want to make here is that there are inherited genetic variants in each one of us on this call that influences how we as an individual will respond to a toxicant. Some of us on this phone call will have actually variants in our blueprint that protect us from a particular toxicant-induced cancer. Some of us will have genetic variants that actually increases our susceptibility to a particular toxicant-induced cancer. And the beauty about the current research, and I will delve into that a little bit later when we discuss some of these topics in more detail, is that for some chemicals, we understand and have identified based on science what these variants are in the different genes that constitutes or determine increased susceptibility, or whether a person actually developed or has a built-in genetic code to protect them for toxicant-induced cancers. A classic example that we typically use in benzene cases are, for benzene, typically it is a non-toxic compound to our bodies. However, our bodies metabolize benzene into a toxic oxygen-rich species, which is known as, sorry, I just quickly forget it, hydroquinones. Hydroquinones are causing DNA damage. Now, we understand exactly which pathways and genes within those pathways, how they control individual response to convert benzene into a toxic compound. Some of those variants are known to actually protect an individual, protect them to convert benzene into toxic compounds. And some of them in specific populations have been found to actually increase an individual's susceptibility. Now, for asbestos, that information is currently being developed. There are already markers that has been identified that can, at this stage, based on specific populations that they've looked at, differentiate between those individuals that are programmed to have a resistance to develop asbestos-induced malignancies, and those people that have increased susceptibility. So, in my opinion, it is that if susceptibility is asserted by a plaintiff's expert, the validity of that assertion can and must be tested objectively. For some toxicants, there is a wealth of information. For some toxicants like asbestos, of course, that is still developing. But for asbestos, as I will discuss later, there are already existing susceptibility or resistant alleles identified that can determine how an individual will respond once they are exposed to asbestos. Next slide. The key of today's talk is obviously to give you kind of an overview of what has recently been published for mesothelioma. And I want to have you at least take these four primary messages as key take-home messages. The first one is, and probably the most important from my perspective, is that mesothelioma is not an exception. Like all other cancers, mesothelioma has now been shown, based on peer-reviewed public literature, that it has specific genes and mutations in those genes that predispose from a hereditary context to malignant mesothelioma. And this is typically can be caused in the absence of a particular toxicant. The second message is that our understanding of which genetic factors contribute to making certain individuals more protective or resistant to develop asbestos-induced disease, and those that are more susceptible is becoming much clearer. And there are based on current publications, some markers that can be used to screen individuals to determine if they are protective or at least at an increased susceptibility once exposed. The third point I want to raise is the fact that we typically assumed that asbestos caused direct interaction with DNA and caused double-strand breaks or single-strand breaks. Research have now shown that the primary mechanism by which asbestos-induced DNA damage is by an indirect response, by activating our inflammatory response. And each and every one on this phone call today is our inflammatory response is governed by our own immune system. And we are unique in our immune response from each and every one. And I will discuss that in a little bit detail later on. The final point here is that there is research efforts currently underway to identify biomarkers, or what people also refer to as fingerprints, particularly for asbestos-induced disease. Research efforts are currently underway to identify mesotheliomas that are induced by asbestos by looking at specific molecular markers. So these are the four primary messages that I would like to have you focused on. And for the rest of the talk, I will go into a little bit more detail in each one of these. Next slide. So for hereditary predisposition to mesothelioma, the first gene that was identified in 2011 was BAP1, or BRCA-associated protein, and it was shown by Testa and Carboni to be a predisposition factor for mesothelioma. In 2016, a paper by Craney actually proposed that up to 20% of all mesotheliomas are initiated by germline mutations in genes that's known to cause the onset of clinically well-defined hereditary familial cancer predisposition syndromes. Up to date, 60 germline genes have been identified in published literature that drives the onset of mesothelioma in a pure hereditary familial component, not just BAP1. If you just screen for BAP1, you miss 59 other germline drivers that's now known to cause mesothelioma. Next slide. Just in 2018 alone, numerous papers have been published that found, and in fact, most of these papers have now identified mutations in genes other than BAP1. A massive study, as Kurt just pointed out, the Panawet paper in August, identified 13 new cancer predisposition genes, genes that are inherited from mom and dad that predispose you to develop mesothelioma. Many of these studies, in fact, in 2018, our understanding of published genes that predispose to mesothelioma has increased from less than 20 to 60 genes that is now known, based on peer-reviewed published literature, to predispose to mesothelioma. And typically, based on pure hereditary inheritance by Mendelian segregation, these mutations are causal. They will cause the disease. When it will happen depends on the penetrance of that particular mutation. Next slide. So the point here is that there are more than just BAP1. There are 59 other genes published as germline hereditary predisposition genes for the development of mesothelioma. And in ToxicoGenomica, we've developed a custom gene panel that will rapidly screen and identify those individuals that are predisposed to develop mesothelioma. And this is a custom panel. It's not commercially available, and it's updated on a bi-weekly basis. And that number, as I said, just in 2018 has dramatically grown from less than 20 to about 60 genes. Next slide. I want to focus also a little bit on ovarian cancer. So ovarian cancer.
Len, do you want me to skip ahead to the ovarian cancer section, or?
So let's go back to this slide. Sorry.
Okay. Yep.
Do the asbestos cancer. So this genetic resistance and susceptibility, that's the important point for us to make. Susceptibility, as I said, there's a lot of studies that's currently being underway that use genome-wide association studies to determine susceptibility or resistance markers or alleles that influence, from a genetic hereditary perspective, how a person will respond once they are exposed to asbestos. Either they are at an increased resistance or they have an increased susceptibility. There are large population studies that's been done in Italy as well as in Australia, where they've identified genes, but also variants in non-coding parts of the genome that actually influence individually a person's ability to be either protective of developing asbestos-induced disease versus those that have an increased susceptibility. For some of these markers, a secondary study with a different population has confirmed, particularly in Italian populations. So as research is progressing, we have and are developing a better understanding of other genetic factors that contribute to a person's ability to either restrict or protect against asbestos-induced disease, and why some people that are exposed are at an increased susceptibility. Next slide. This brings me to the point of our understanding how asbestos induced damage to DNA. Initially, it was thought that DNA directly interacts with asbestos and causes breakage points like that. However, it's now been shown that chronic inflammation, our own system is activating an immune response that ultimately create a reactive oxygen species. And these reactive oxygen species then interact directly with DNA, causing these damage points. There's a very a nice review paper published by Sage et al. Just very recently, which describes this mechanism in detail. So next slide. What we now understand is that our individual response to asbestos are not only determined to other genetic markers that is present in coding parts of the genome or present in non-coding parts, but that our own immune system and the genes that drives our immune response determines our ability to react towards asbestos exposure. We now know that the genes that drives our immune system, there are cytokines, 19 different genes being published, and that variants within those genes dramatically impact how each of us activates our immune system that directly activates our inflammatory response. The point I want to make here is that 75% or up to 75% of the genetic variants that determines our immune response is inherited from mom and dad. And that clearly indicates why some people are more resistant to develop asbestos-induced disease than others based on how they activate their own immune system and how we respond by our inflammatory pathways towards exposure to asbestos. It's uniquely individual, and what drives that are now expanded to genes involved in our immune system activation. And that is added to the genes that we have identified thus far in genome-wide association studies that make some people more resistant to asbestos and other people more susceptible. Next slide. The last aspect for asbestos-induced disease is the research that's currently focused on identifying biomarkers or fingerprints that differentiate between cancers that was caused by a toxicant, in this case asbestos, and those of the same cancers that was caused by other etiology. One of the key focus areas are microRNAs. Now, microRNAs are non-coding non-coding genes, so they don't code for a protein, but they are known to be regulatory mechanisms. They are basically the master puppeteers controlling switching on, switching off suites of genes. Now, next slide. A paper that was recently published by Santarelli, literally 2 weeks ago, came out with a fascinating fingerprint that they deemed, based in their conclusion, as a signature that could have major medical legal impact. What they basically did is screened for people that develop asbestos-induced mesothelioma and asbestos-induced lung cancer, and they had a population of people that developed the same diseases, but for which there was no a known exposure. What they then did is that they identified a series of 4 microRNAs and how they are expressed in these different individuals. They then tested that microRNAs in a separate population and confirmed that they are indeed able to separate out between those diseases that were induced by asbestos and those that are not. Then they looked at a third population where they actually had a blind validation, and they determined with very specific sensitivity and specificity values that they were accurate in predicting and identifying individuals that developed mesothelioma due to asbestos and those that did not. And the same for lung cancer. Now, there are limitations, and I clearly want to point that out. This is based on a relatively small study population. And then, of course, the ultimate factor is our difficulty to estimate asbestos exposure. But nonetheless, studies are currently underway to identify molecular biomarkers of fingerprints that ultimately would allow us to differentiate between those diseases that were directly involved in activated by a toxicant, in this case mesothelioma, and those that are not. Next slide.
So I just want to pause here for a brief moment, and I just want to make it clear that if anybody has a question, and you can do them in real time if you'd like, if you think of them, you can type them into the chat menu in the GoToMeeting sidebar. So I just wanted to make that clear, and we have some questions already. And Len, I'll let you continue with the ovarian cancer section.
Okay, next slide. So for ovarian cancer, I have a similar series of take-home message, but I think the most important aspect for ovarian cancer is ovarian cancer is one of the most genetically predisposed cancers, much more so than what we currently estimate or know for mesothelioma. There are currently more than 30 genes, in fact, 37 genetic drivers that's been identified to predispose to ovarian cancer, other than BRCA1 and BRCA2. Next slide. This slide demonstrates why and showing that certain cancers for ovarian cancer is predisposed is actually in this study from 2015, 19% of all ovarian cancers are characterized by a pure hereditary predisposition compared to other cancers. Next slide. In fact, by a paper published in 2015, Testa et al. Concluded that 23% of all ovarian cancers are due to a wide range of inherited driver mutations. I want to highlight one particular paper here, and that's the Phelan et al. 2017 study. It was published later in 2017. This was a massive study. They, in fact, sequenced over 100,000 women, sequenced the genomes for those 100,000 women across the entire world. I think more than 100 different laboratories participated in this study. And they identified at the end of 2017 12 novel predisposition loci for ovarian cancer, specific to ovarian cancer. So it's not just BRCA1 and BRCA2. Many studies, next slide, even in 2017 and 2018 alone, has been shown that additional mutations that have never been found before to be associated with hereditary breast and ovarian cancer, in particular breast cancer, have now been shown to predispose. We are currently at 37 genes that are known to predispose to hereditary ovarian cancer. Next slide. What's very interesting from a research perspective is that, as I pointed out in the beginning of my talk, all these hereditary cancer predisposition syndromes, of which hereditary breast and ovarian cancer is a well-known one, has clinical inclusion criteria developed. Now, these clinical inclusion criteria are used by physicians or by a medical specialist or genetic specialist to identify those individuals that most likely are predisposed to a particular disease. And they develop these criteria not only clinically, but also based on family history. But the study that was published by Rowley et al. In 2018 found that although these clinical genetic testing is very well accepted and the inclusion criteria is very well established for hereditary breast and ovarian cancer, that many people, women in that population that they sequenced, was found to be positive for known hereditary predisposition mutations that predispose to the development of ovarian cancer, but that would not fulfill the inclusion criteria developed for ovarian cancer. This implies not only that even for a well-studied cancer like ovarian cancer, there is still room for improvement, but it further highlights the importance of why people need to be sequenced. Only once you've sequenced the genome, either the germline or the somatic tissue, the cancerous tissue, can you and will you understand what drives that tumor. And what their study found that even in today's highly developed criteria for hereditary breast and ovarian cancer, that many times those women were found to have genetic drivers, but they were not included or would have not been selected from based on their genetics due to family history. Next slide. So for ovarian cancer, I want to highlight the point that we've developed a custom hereditary ovarian cancer predisposition panel as well. I want to point out that you cannot just focus on BRCA1 and BRCA2. There are numerous other known germline drivers for the cancer, and that we are constantly updating our custom panel to reflect the latest published data. And this is obviously also ever expanding. In fact, 2 months ago, there were only 32 of these hereditary drivers known for ovarian cancer. We are now at 37. Next slide. So one of the things that I kept mentioning is the custom panels that we've developed. When we are presented with a toxic tort case, we have 3 different levels to look at for genetic testing. Now, what I preferred as a scientist is to look at the full genome. And typically, the full genome gives you a lot of information with regards to other aspects of coding and non-coding parts of the genome that is very important in rendering an opinion as to causation. But since we are bombarded with so many cases in ovarian or in mesothelioma, we decided to develop a tiered approach. Next slide. And the tiered approach for us is basically looking at first of those very well-defined hereditary predisposition genes, 37 for ovarian cancer and 60 for a germline predisposition to mesothelioma. And if we don't find any of those germline predisposition genes, we expand the panel to the next tier, which is termed the broad cancer specific panel. Here we look at all the genes that has been published up to date in both germline and somatic contexts for both mesothelioma and ovarian cancer. For ovarian cancer, this panel contains 488 genes at this stage, and for mesothelioma, 2,508 genes. And then if we don't find any of the drivers in those genes, we then expand it to a pan-cancer panel. A pan-cancer panel basically consists of 3,500 genes, and these genes are genes that's been published to be involved in either the initiation, tumor development, and metastasis of all cancers. So what we're trying to do is that we keep building on the knowledge. If we don't find it as the low, high impact ovarian and meso panel, we can expand it to include more and more genes. We also know that mesothelioma, for instance, is now closely associated with melanoma, breast cancer, pancreatic cancer, kidney cancer, and lung cancer, meaning that they share similar genetic drivers. And many times we don't pick these things up when we do these focused panels, but when you expand it, you can identify pathogenic drivers in the pan-cancer panel that is known to be associated with another cancer, but has been shown to be similar or also known as a driver in mesothelioma. So it constantly pays to kind of develop and expand our research efforts. We produce a report for each one of these. All of these service levels come with a basic report where we basically determines absence of presence of particular variant. We can then provide this report to external experts that can render opinion as to causation. But typically we proceed with our own expert reports in these cases. We have two levels. We have a curated report, which is basically where we start linking the particular mutation that we found in an individual with published literature as to the initiation or onset of the specific cancer or related cancers. Or the final level comprehensive scientific report is when we actually go down into the mechanistic level of why this person developed this particular cancer and this particular pathological subgroup, because we understand which biochemical pathways are involved, and mutations within those genes in those pathways can be intensely studied with supporting literature to develop a mechanistic understanding in depth from a scientific perspective how this person developed this tumor specifically. Next slide. I want to leave with, go on to the next slide, David. I want to leave with some specific criteria that you as defense counsel can be on the lookout for that actually point or act as red flags for which cases are most likely genetically predisposed. Now, it's incredibly important from a genetic counseling perspective, as well as a geneticist, that we've developed, as I said, these clinical inclusion criteria for all these different types of cancers. Now, when you average out all those inclusion criteria, you come up with this list of criteria that can be a guiding factor as to guide you very quickly whether the new case could have a genetic predisposition. Family history is very important. If you see multiple cancers in either maternal or paternal lines of the family, that's usually a strong sign that this family might be one of those families that are known to be genetically predisposed to a particular type of cancer. Even more stronger evidence if first-degree family members present with the same or related cancer. Say, for instance, as mesothelioma, which we now know includes breast cancer, includes pancreatic, kidney, lung, and melanomas. And if you have first-degree family members like mom, dad, or siblings that have related cancers, that's an immediate red flag that something from a genetic perspective is most likely driving tumorigenesis in that individual. When you look at the personal medical history, as Kurt pointed out early, when a person develops cancer below the age of 50, that's an immediate red flag because cancer is typically a disease of the aged. So when a person presents with the early onset cancer, that should serve as an immediate red flag that there could be a genetic underpinning to that disease. In most of these cases, in fact, the Panau paper concluded that they identified these germline drivers in people that are primarily very young, people that develop secondary malignancies, and people that are typically not associated with asbestos exposure, particularly in the Panau paper for genetic predisposition drivers for mesothelioma. The same is true when a person or a plaintiff present with secondary primary tumors, either diagnosed before ovarian or mesothelioma or later in life. That's a very clear sign that there is something else going on in that individual, and that's most likely an inheritance pattern of highly pathogenic drivers. Thank you, guys. Kurt, can you do the concluding comments?
Sure. So we've thrown a lot of information at you in a hurry, so we want to try and return to big picture points. As we see more and more mesothelioma cases that involve low-dose claims, often take-home claims, it's not terribly surprising that plaintiffs' lawyers are embracing susceptibility arguments because they have very low exposure evidence, if any, to point to. So, at least in our view, this is going to continue. It's been seen in some of the cases, including Ingham and the chart that we showed you earlier that was used by Mark Lanier to argue the ovarian cancer issues. So our suggestion is that people need to know that's out there, take it on both in depositions and at trial, and consider using some of that with the treating physician and show their lack of knowledge. And we also would urge you to keep an eye out for the mouse data, which is going to shape some of the future arguments. We're watching for that. Those of you who are taking depositions will have an upfront chance to ask about that. At the broad level, our view is the defendants need to change the paradigm. Don't let plaintiffs' lawyers appropriate and misuse and oversimplify this notion of susceptibility. As Len said, it's a complex concept and it's being misused, and it should not be misused. You want to focus instead on the fact that hereditary and familial predisposition cancer syndromes are very real things. Genetic counselors advise people to make life-changing decisions based on the presence of genetic mutations, and these are diseases that are arising because of genes, not because of exposure. So take the focus and reframe it onto the fact that most people are resistant and that cancers are in fact going to show up in some people simply because of a genetic predisposition. We also think another virtue is that you can use the data to give the jury something instead of having to argue idiopathic, and that prevailed in the Guzman case in Louisiana. So we think that really ramps up an alternative cause argument. We also want to point out that sequencing is becoming much more cost effective, especially if you can run multiple samples at the time, and we've done our own work and custom gene panels to facilitate that. I also want to add one comment about the fingerprinting efforts. This is ongoing. It's not here and now today, but do be aware that these efforts are out there and are another reason to keep an eye on what's happening in the science. Some of you may recall that there were already efforts to fingerprint, if you will, a signature for asbestos-induced lung cancers, and that work on this topic was presented and discussed at the Helsinki conference in 2014. So this is a topic that's out there, and as the molecular tools continue to improve, you'll see more efforts along this line. Ultimately, it can be both good news and bad news for defendants, depending on how things turn out. So with that, we want to open it up for questions.
Yes, okay. So we have one question here. It says, have there been any decisions allowing BRAD-based genetic testing? I'm not sure BRAD-based. I was trying to figure. Genetic testing seems like it would be an uphill to test for more than BAP1 or maybe some others in a case. Does staining the tumor for the mutations a good first step to persuade a court to allow genetic testing? So I guess the gist of the question, and Kirk, maybe this is a good one for you, the gist of the question is, is it an uphill battle to sort of convince the court to let you do genetic testing?
It's correct in general that most people have pitched these in the context of BAP1, but I would not limit it there. I would not encourage staining in advance, where that did happen in the Ortwine case. You happen to have Dr. Carbone involved, who's far more knowledgeable than most. I think there's sufficient precedent out there now that you should be able to prevail on a broader request for genetic testing. And papers such as the Pinal paper, et cetera, show that it should not be limited. A few years ago, perhaps it was appropriate to only look at BAP1, but today we're well beyond that. And hopefully judges are reaching that conclusion. We can also point you to non-asbestos cases where broader genetic testing has been done and gene expression testing of the sort that was used in the Guzman case. So I would urge you to take a broad view of things.
All right, we're getting some good questions here. Next question, is blood testing of the primary plaintiff all that is required, or must you get blood testing of all family members? What are the time requirements to doing this testing? Len,
and let me take that on to start and give the simple answer. Len may want to amplify.
Sure.
In general, the best way to get genetic testing done is with a small sample of fresh blood and then shipping it in cold storage. It's not terribly complicated. That will provide you with all of the information about the plaintiff themselves. Whether you want other and further information and to seek some other genetic testing would, in my view, depend on what you have in the way of other discovery. If there's a strong track record of familial cancers in different relatives, I would suggest you don't need to go and ask for additional testing, and you will find that a little more difficult. However, there are cases where courts have permitted testing of other family members. It may or may not help you depending on the specific case facts, is my view.
If I can add to that, like in the Jessica Blackford-Clayton case, the treating physician has recommended genetic testing of both the plaintiff as well as her family. So when we received the medical records, genetic testing was done at both with the plaintiff as well as with the mother of the plaintiff, and has demonstrated that there was a direct inheritance from both, from the mom to the daughter, in a Mendelian segregated manner. So in some cases, the medical records, with the prevalence of genetic testing being commonplace now, many times you will have multiple family members already being subjected based on an extensive family history. Another point, so you might have that data already available. Another point that's very important, many of the cases that we are involved in, particularly on mesothelioma, is when the plaintiff has already passed on. So you cannot get blood. And in that case, we go back to the medical records and we look for particular clinically fixed or preserved tissues. During the lifespan of most individuals, there are biopsies done, and when a pathologist renders those biopsies as cancer-free, we can access those and extract viable tumor-free DNA that can be then used. We've never had any issues for the court to actually get us access to clinically preserved formalin-fixed paraffin-embedded tissues that can be used to identify germline drivers. So it's not only blood. In many cases, we can go back in the medical records and identify non-cancerous tissues from past medical procedures.
And the formalin-fixed materials can be used many, many years later.
Oh yeah, 20, 30, 40 years. We worked on many old. Usually they are subjected to be kept 5 years, but in our experiences, we've accessed tissues that are 20 years old, and you can get very good, high-quality DNA from that.
All right, this next question is a really nice synthesis question. Can a person have a genome which makes him both predisposed and susceptible to mesothelioma? So a great question. I think it's insightful. It sort of synthesizes, and the short answer is yes. Len, why don't you say the
yes. So this is particularly true in case with BAP1. BAP1 is the only cancer predisposition gene that has now mechanistically shown to also make a person more susceptible when exposed to asbestos. So not only do we know that BAP1 particularly predisposed, it is now the latest member of it's now the 201st hereditary cancer predisposition syndrome. BAP1 has been classified as its own genetic cancer predisposition syndrome. So we know it can cause up to 17 different cancers. However, what's really interesting is that the study that was done by Bononi, a published study last year, that showed if you are a BAP1 germline carrier, you are also increased susceptible to asbestos, even low-dose asbestos. And they described mechanistically, due to calcium flux outside the nucleus, how that increases a person's susceptibility. So the answer is yes. You can have an individual that have a genetic predisposition to develop the disease, but also have an increased susceptibility.
And if I can just add to that for a minute, Len, it really highlights the importance of looking much broader, because this would be a case where if you just had information on BAP1 and the health of the BAP1 gene, the plaintiffs might be in a position to argue susceptibility, but if you had a broader sense of exactly what the BAP1 mutation was and the context of other mutations in that individual plaintiff, you would be in a much stronger position to argue a predisposition syndrome. So it's really, it really highlights the importance and the short-sightedness of just looking at a single gene.
And then just to add on, it also takes you to the point of framing up this issue of predisposition versus, quote, susceptibility. And there is a tremendous amount of genetic counseling literature out there. And at least in my view, one thing that defendants need to do more of is to show this is not just litigation-related science. This is science that is applied in daily lives of people making dramatic decisions. And Len makes a point of incorporating that information where it's available as to a specific cancer. And that helps to frame this issue in a more positive light that does not sound so litigation oriented. And then further on this question, we're going to see evolving information as Testa's mouse studies come out. And in case I said it too quickly, I do want to emphasize that they are doing more knockout mice studies, meaning they are altering the genes in mice and then exposing them to asbestos fibers, including chrysotile. And then people are going to make different arguments based on whether those mice end up developing mesothelioma without being exposed to fibers. A couple of mice have already done that. Now, some people will go ahead and say that's not enough, and it's going to change the dynamic here because some defendants may not be comfortable pointing to animal studies because lots of defense lawyers have spent years opposing those. That's why it's important in our view to keep an eye on what's happening in this area and to know new data will emerge.
It's important to note that mice, that the medical progression or the pathological progression of tumorigenesis of mesothelioma in mice are almost 100% identical to what happens in humans. Mice are considered to be incredible models for studying the initiation and progression of tumorigenesis in mesothelioma. And that's why, from a research perspective, we're very keen on the idea of looking at mice because you can have absolute controls. With humans, you can never exclude asbestos exposure. Even for us that are sitting here, we all are exposed to some level of asbestos. But with mice, you can have absolute control. And then that's where you can really start identifying biomarkers for exposure, specific asbestos-induced disease, etc. And that's why mice, from our opinion, are excellent models, and that's why Carboni and Testa and all of those people are really focusing on using mice to develop a clear understanding of what drives and initiates tumorigenesis in mesothelioma.
All right, everyone. Thank you very, very much for your time. We hope you found that informative, and please feel free to reach out to any or all of us with any additional questions or if you'd like to chat about the role of genetics in litigation. All right. Thanks very much.
Thank you, guys, for your time. Appreciate that.
Thank you.